Okay, so to pick up where we left off, we're going to be talking about the components to cardiac tissue and the cells and their main functions. Okay, let's start with the SA node. The SA node is a cluster of pacemaker cells. This operates by sending signals at least 60 to 100 cycles per minute. It is located up in the posterior wall of the right atrium. So it makes sense that the right atrium depolarizes slightly ahead of the left atrium. So that first half of that P wave we just talked about, how the right atrium is depolarizing slightly ahead of that left atrium, where the left atrium is the second half of that P wave, makes sense because the SA node, which is automating our cycles per minute, sending those signals, is located in the in the upper corner of the right atrium. Okay, the AV node is the second cluster of pacemaker cells. This is located between the right atrium and the right ventricle. So we will send a signal down through this so that the ventricles can contract / depolarize as a unit, right? Pretty much the same thing. So we need time for the atria to depolarize first, okay? So if the signal was received in the ventricles instantaneously before the atria depolarized, the atria would not be able to complete their depolarizing before ventricular depolarizing was initiated. So we want a little bit of time for the atria to depolarize first before the ventricles. Okay, the atria forces another 20% of blood into the ventricles when it depolarizes first. So this is referred to as the atrial kick, or that atrial contraction. So this is why we want the atrium to depolarize first, right? So they can slightly depolarize and contract, shooting an extra 20% of blood down into the ventricles before the ventricles all contract, and they send the blood out into either the pulmonic system or into the systemic system. Okay, so continuing down, we're going to talk about the bundle of His. So this slows everything down for us, right? So there's a couple bundles, but the bundle of His is located a little bit further down in the heart. So this slows everything down. This comes off of the AV node and slows down the electrical current to give time for the atria to depolarize first. So this is when the signal gets shot down from the SA node through the AV node. The bundle of His is actually going to slightly slow down that electrical current in order for the atria to depolarize and contract, give that atrial kick of the extra blood into the ventricles, and then continue on with the electrical signal, signaling the ventricles. Okay, so continuing with the next step, after the atria has dumped their contents, then the current will pick back up again, go down the bundle branches to the Purkinje fibers. Okay, so we have the SA node, we have the AV node, and then the His bundle is coming off of the AV node, right? Okay, it's going to slow everything down so these can contract first. So the electrical current needs to be at a great speed now because we want the ventricles to contract as a unit. So after it goes through that bundle of His, it's going to speed back up again. But again, we want to contract these all at once. We don't want the left ventricle to contract to half a second before the right, and all of this tissue. So we want all of this tissue to be contracting together. We want them to be contracting as a unit. And that's how we're going to be more efficient and send the most blood out all at once. So, however, this is our ventricular septum. Remember from the anatomy of the heart we talked about. The septum will always contract first because of the presence of these fascicles, which are also considered the left bundle branch or the right bundle branch. So fascicles represent avenues for the passage of electrical current. Okay, so let's talk about the fascicles. The right fascicle is in the right ventricle and is called the right bundle branch. So if you see the terms right fascicle or right bundle branch, it's the same thing. This stretches into the Purkinje fibers. The Purkinje fibers are along the outer side of the heart, which is going to continue that electrical signal. So the right ventricle only has one fascicle. So do we see here we have one right bundle branch, which is one fascicle. Right? Because the right side of the heart does not have as much tissue to depolarize. So we talked about that before, and we talked about, this is going to all come into play as when we talked about the pressures of the side of the heart. It makes sense Do we have to contract as hard on the right side of the heart to send blood just to the lungs? No, versus the systemic side. So all of this kind of is adding up and makes sense. So remember, it is a low pressure system on this right side. So we have one right bundle branch, and that means we have less tissue on the right side to depolarize compared to the entire left side of the heart. This is why I wanted you to consider it a left side and the right side of the heart. Okay. The left side of the heart, this is a high pressure pump to the systemic circuit. So we have 3 fascicles on the left side of the heart to help overcome this large pressure system and pump blood into the systemic circuit. Okay, so we have a posterior fascicle, an anterior fascicle, and something, in the left bundle branch right here. So we have 3. Okay. Okay, the first we're going to talk about is the septal fascicle, okay? This is the first to contract or depolarize. We need a rigid septum so the ventricles have something to contract against, right, to push up the blood up through the heart. So that makes sense that we would need a rigid septum in the middle of the heart. This is the first portion of that QRS complex. So remember, we had the Q, R, and the S. That Q wave is the septum depolarizing. So let's go back to look at the anatomy of the heart, and here's the septum. So what's going to happen first is when you look at these different bundle branches, the septum is going to depolarize first, so then it becomes a rigid septum, so then the ventricles can contract against this septum and push the blood out with force out of the heart. Okay, now the other 2 fascicles on the left side of the heart, we have one fascicle is the anterior portion of the left ventricle, and one fascicle sends a signal to the posterior portion of the left ventricle. We have to have 2 because there's a large amount of myocardial mass on the left side of the heart. We need these 2 fascicles to depolarize, contract at the same time. So that's why this electrical signal has to go through pretty much all at once. And remember, we talked about the cells, what it can go through. We have electrically conducting cells that have the presence of gap junctions, and that allows pretty much a resistance-free environment. So you can see all of this stuff is kind of tying together with the function of the heart. Okay, so now we just told about what sections of the heart are depolarizing from an anatomical standpoint. Let's look at the ECG. So if we look at this Q wave, let's look at just here. Let's look at this QRS complex. The Q wave is the septal fascicle has fired, right? So the septum has fired there, so then we're going to get that signal. But now it's just the septum that's depolarizing. So it makes sense that this waveform is a little bit smaller because the septum itself doesn't have that much mass, right? Now the R wave are the other 2 fascicles firing, okay? So those are the anterior and the posterior portion of the left ventricle, this R wave. You could see how much muscle mass that has to depolarize, that electrical current's got to go through, right? So there's a large amount of muscle mass on the left side of the heart. And then the S wave are the Purkinje fibers depolarizing, right? So it's the outer portions of the heart over here, right? So we got our Purkinje fibers that go to the rest of the heart, okay, on the outside over here. And then they're over here. So the S wave is the Purkinje fibers. So to review what we talked about before, let's go into what segments are. This is points of electrical neutrality. So we have a segment here. It's our PR segment, okay? So this means no waves are included, right? So then electrical neutrality occurs here. This ST segment, anytime you hear that a segment with regards to ECG, it represents electrical neutrality. Okay, so now let's review an interval again. This PR interval. An interval is some sort of segment plus a wave in form. So it's some sort of, it represents electrical neutrality as well as some sort of depolarization or repolarization of tissue. So let's look at our PR interval. PR interval is going to be the atria depolarizing, contracting, as well as some electrical neutrality before the ventricles depolarize and contract. Then we have a little bit of electrical neutrality, and then we have our ventricles repolarizing. So here we have the atria depolarizing and some electrical neutrality. That makes sense because what occurs here after the atria depolarize? We slow down at the bundle of His, right? So and we want that so we can get that atrial kick so more blood goes into the ventricles. Makes sense that we slow down right here. We have electrical neutrality. Then the ventricles are going to depolarize. Okay. So the QT interval, let's look at this full QT interval, is the ventricles depolarizing. We have some sort of electrical neutrality, and then they are going to repolarize. So we have a wave segment and a wave. So we have here, so if we want to talk about the QT interval as a whole, we have a depolarization of the septum, depolarization of the left ventricles, depolarization of the Purkinje fibers, and of the ventricular wall, some electrical neutrality, and then we have ventricular repolarization. Okay, so let's talk about what ionic movement is. So all of these events that we just talked about and what they represent on the ECG, all they represent is ionic movement. So some waves are positive and some waves are negative on the graph. Why? So this is due to the relationship between depolarization and repolarization and the placement of an electrode on the body. Okay, so anytime there is a moving wave of depolarization going towards a positive electrode, you will have an upward deflection of the stylus. When you have a downward deflection of the stylus is when the moving wave of depolarization is moving away from a positive electrode. Okay, so I'm gonna say that again. So let's talk about the depolarization in general. Anytime there's a moving wave of depolarization going towards a positive electrode, you will have an upward deflection of the stylus. When the moving wave of depolarization is moving away from the positive electrode, you will have a downward deflection of the stylus. Okay, so that makes sense. Going something towards positive, you're going to have an upward deflection, a positive deflection. Anything that's going away from something positive, you're going to have a negative deflection. Now that's depolarization. Now repolarization is going to be the opposite. Anytime repolarization goes away from a positive electrode, you will have an upward deflection. Okay, and anytime repolarization goes towards a positive electrode, you will have a downward deflection. Okay, so it's just the opposite. Depolarization and repolarization are opposite, and it makes sense when you think about the layers of the tissue that are depolarizing and repolarizing, and which we're going to get into in the next slide. Okay, so on the next video, we are going to pick up here with talking about the layers of the heart and how depolarization and repolarization play a role with the upward and downward deflection of the stylus based off of where the signal is moving through the layers of the heart.